This edition of the Bio Report is brought to you by the California Technology Council, providing discounts on products and services essential to every startup. For more information, visit californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. CellMax Life believes it can transform cancer diagnostics with its affordable, non-invasive test for early cancer detection. The company says with its platform, it has been able to overcome the challenges of detecting rare circulating tumor cells in precancer and early-stage cancer patients when the disease can be successfully treated. We spoke to Atul Sharan, co-founder and CEO of CellMax, about the challenges of liquid biopsies, how the company's technology works, and why he thinks it has the potential to change the way cancer is diagnosed today. Atul, thanks for joining us.
Yeah, thank you, Danny, for having me.
We're going to talk about CellMax Life, its liquid biopsy test, and the promise to detect cancers at early stages with simple blood tests. I thought you could begin by discussing how CellMax came about, since you were an entrepreneur with a history of building companies in the high-tech world of Silicon Valley as opposed to the life sciences. How did you become interested in early detection in cancer?
Sure. It's actually quite interesting because about 5 years ago, my wife in the Bay Area, she went through a routine mammogram and she was qualified as low risk. So it was a very routine mammogram. There was nothing to suggest that she was any higher risk of breast cancer than average. And, the mammogram was actually clean. So she got a letter from the, clinic saying, congratulations and, come back and see us next year. By this time, I was resident at a venture capital firm, and a little coincidentally, one of the issues we were looking at was, the outcomes in cancer not having changed, pretty much for most cancers in the last 50 years. So and this is true, and I, against the advice of her longtime physician, pushed her to get more imaging because I had looked at the numbers, and, the mammograms are a great test for some women and not so great for others, and especially if you have, dense tissue. So she reluctantly went for further imaging, and long and short of it is, within 2 weeks, she was diagnosed with, early breast cancer. Now, you could argue that the screening didn't work and the system didn't work, or you could argue that it did work in the context of the consumer, the patient taking control, of their health and decision making. So it led us to sort of form the thesis that if, when you look at the numbers, the outcomes of cancers have changed where there's simple, affordable screening tests that can detect cancer early, but where the compliance is high. And then when you start looking at the problem globally, this is even more stark. The second thing I should mention, Danny, is that, my mother also passed away of esophageal cancer, and that was, discovered very late stage. So the other problem you ask yourself is, why are the majority of the people in most cancers detected late stage? And the reason is that cancer is asymptomatic. So unlike even the flu, you don't have any symptoms. And a lot of times, for example, in colon cancer, the symptom will be when the colon is actually, blocking the tumor is blocking the colon. So it'll be like an indirect symptom, right? So for something like that, it is even more important that there be, an affordable, simple screening test. So that was the sort of genesis of, getting started.
And what's the problem exactly that you were trying to solve? And is there something in the approach of your platform that speaks to your engineering background?
Yeah. If you look at screening tests or early detection tests, they're all different modalities, right? There's like low-dose CT scan for lung cancer. There's PSA, for prostate, which is really not even a cancer marker. And there's, of course, mammograms for breast and various other things. And the reason is that, as you know, which is also changing, that cancer has always been thought of as a disease of the organ when, in reality, it's really a disease of the genome. So one of the fundamental, and this has been known for a long time, mechanisms of metastasis is circulating tumor cells. Okay, and these, it's actually very simple. Nobody really dies of the primary tumor. They die of metastasis. So if you can find the primary tumor early on and resect it, there's no debate that that's your best outcome. Now, I will say that there is interest in the medical establishment, especially in the drug companies, to maybe counter that. But nevertheless, it's been well known that these tumors in the very early stages, they shed tumor cells into your bloodstream, and hence it's called circulating tumor cells, or CTCs for short. And your immune system will try to kill them, but sometimes it's successful and sometimes it's not. So people have known this, and people have known that if we can, via a blood test, detect these CTCs very early, that could be the first indication that something's wrong, and then you can figure out what to do from there. The problem has been it's like looking for literally a needle in the haystack, right? So, and the advantage of being at a venture capital firm is that you can get to look at all various technologies available, and at least try to see which ones might work. And because of my entrepreneurial background, I was interested not just in finding the technology, but also building the company. So we looked around, literally all over the world to identify a technology for CTCs that would, be able to detect cancer early. And surprisingly, it was kind of in our own backyard in the Bay Area, and it was not. It was almost 5,000 miles away. So there was a researcher at Stanford who had been working on this problem, and I think in the early 2000s, she was then invited to go back to Taiwan, where she was originally from, because they had started in a very aggressive biotech program that the government was funding. It was started by their only Nobel Prize winner, Y.T. Lee. So she had started doing some studies with the hospital there that showed evidence of early cancer detection. So we ended up, basically getting the rights to that technology and starting the lab, in Asia and in Silicon Valley. Now, regarding your other part of the question about the engineering background, so one of the things that, I grew up in the semiconductor industry. I came to, Silicon Valley in the early 80s when there was really silicon. There was no software. I don't think the word genomics was even in parlance at that time. One of the remarkable things that the semiconductor industry has done, and when I was here, it was, fairly manual process actually, is, driving the cost down through automation, right, of both, mechanical, electromechanical automation, if you will, the automation of the chemistries, of the software, of the chip design, everything, right, which then allowed them to, decentralize it and manufacture it overseas. And a lot of it was based on standardization. So one of the philosophies was that the assay would have to be first tested to make sure it works properly, and then developed with an eye to automation so that it could be automated to offer, the test at affordable prices.
For people not familiar with liquid biopsies, can you explain the idea behind them?
Sure. I think like you pointed out earlier, liquid biopsy essentially is, instead of going to the tissue, in the tumor, instead of interrogating the tissue, you get to interrogate the signals that the tumor is giving in the blood. Now, the reason that this is very fascinating and attractive is because, as people have learned over the last few years, in fact, not that, quite recently, the tumors are heterogeneous, okay, which means that, if you are going to do a biopsy on a tumor, there is good likelihood that you might miss parts of the tumor that are substantially different. That's number one. Number two is, and this is the other sort of, it's not a secret, but maybe not that well known, that when somebody says that they found a tumor, what you don't know as a person is that they did not find other tumors or other problems that you might have in your body. So if all of these tumors are shedding signals, let's say, for now, into the bloodstream, and you have the ability to pick up the signals, then, from an engineering background, you would call it like a systems level thing, right? So you're getting a system level signal from the human body as opposed to, a micro point. Now, it looks attractive, but unfortunately, it's sort of been bastardized a little bit to mean just circulating tumor DNA, which is just one of the markers. And in fact, for early detection is too late because it really does come, from circulating tumor cells. And the reason that you will see liquid biopsy unfortunately become synonymous with ctDNA is because Illumina is a great enabler for driving NGS. Okay, so you can take a drop of blood and you can, look for the circulating tumor DNA and you can sequence it. But in reality, what you need is a multi-biomarker approach, and especially for early detection. So you need to look for, cells, you need to look for ctDNA, and you need to look for RNA. You need to look for all kinds of signals that might be, in the blood and not just fixate on one of them. So a true liquid biopsy, which is what we do at CellMax, would indeed do that. Which is why, Danny, uniquely, at least we believe, uniquely allows us to do early detection. Now you'll see a lot of liquid biopsy companies based on ctDNA that, are only sort of relevant in, like stage 4 or very late stage.
So in liquid biopsies, I understand them. The real challenge is being able to identify that circulating tumor cell in a large volume of blood. But are you looking at other things, or are you finding those tumor cells? What exactly is your technology doing?
Well, we're looking. So you bring up a good point, because we are looking for tumor cells, and literally in one drop of blood, there's going to be like 1 billion cells, and only maybe like 5, CTCs in an early disease patient, right? But and you bring up a good point, which is that once you find the CTCs, and the way our technology works is we can actually get these cells intact, and then they can be, interrogated for proteomics and, RNA and DNA and everything else, which actually becomes quite valuable for, not just for diagnostics, but for drug development as well.
So how exactly does your platform work?
So our platform, the innovation is sort of layered on many years of work that the industry had already done on, microfluidics. So we have, a proprietary platform which uses proprietary antibodies and uses the epithelial properties of cells to find these cells. So there's a lot of, you were talking earlier about the sort of the high-tech world, and there's a lot of multidisciplinary expertise that goes into this. So it's, it's chemistry, it's mechanical engineering, it's chemical engineering, it's biology. And we have some material innovation with which we coat these microfluidic chips, which have proprietary, almost nanoscale patterns on them. And the blood, 2 milliliters of blood will flow through this chip that we prepare with these proprietary coatings and, antibodies. And that will trap, the circulating tumor cells of interest. And then we have proprietary mechanisms to release those cells intact onto a membrane, so they can be, further interrogated. But they can also then be basically what's called stained, using the same protocols that you would, use for tissue, to then identify, what kind of cancer are you talking about. And we can do all of this. I would submit to you at an order of magnitude less than what, companies are trying to do by looking for ctDNA through ultra-deep sequencing.
And what's the business model? Are you a CLIA lab? Do you sell directly to doctors, to patients?
So, there was an article in The Economist quite a while ago, actually Harvard Business Review, that said that the hurdle for innovation, medical innovation, to enter the market is the highest in the U.S., okay? And yet, that innovation doesn't seem to help consumers because, the drugs and everything else, the costs seem to be going up. I forget what exact number is, but it's, the healthcare is one of the biggest sectors in the percentage of the GDP. So one of the thoughts or the strategies or the approaches from the beginning was to get the technology to the consumer in a timely manner and not let, that be prevented for either cost reasons or regulatory reasons, but of course do it in a very legal manner. So, the simplest way to do it, which has been enabled, is through the CLIA lab accreditation. So we are CLIA and CAP accredited lab in the U.S. and CAP accredited in Asia, and we want to offer these tests, and we are offering these tests in Asia right now at sub $500 or sub even $400, and we want to enter the market in the U.S. with the CTC test this year. And, we can certainly go much lower than that because we've already been automating it. So in parallel, we can pursue, insurance reimbursement and other things. But we feel that we can actually get this to the consumer without waiting for, reimbursement because it's going to be a highly affordable test. The model which you're seeing from, sort of the NGS consumer-oriented companies is called consumer-initiated, physician-mediated, right? So the consumer says, they want the test and they are willing to pay for it because it's highly affordable, sub $150, let's say, and then the, physician just orders the test, and because it is a CLIA lab and the test can be administered. Now, I can tell you that we've been more obviously active with the CTC tests in Asia, where we are listed in a lot of the hospitals in some of the countries, as official tests. But overall, the strategy is self-pay, affordable, and be able to bring this technology to help the consumers and the patients fast to market.
When I've spoken to other companies working on liquid biopsies, they seem to be focused on using these tests to stratify patients, make treatment decisions, and monitor patients for remission, patients in remission for recurrence. You're looking at this as a diagnostic tool to detect cancers early. You've recently announced a new study supporting that use in colorectal cancer. What did it show, and what do you know about the accuracy of these tests?
So let me get to the first question just for completeness, which is the one you're talking about on the disease side. So we do that too, right? So if you look at it at a higher level, if you can detect the extremely low signal, difficult problem in early detection, then you can certainly apply the same technology for, minimal residual disease monitoring, recurrence, and we do that as well, right? And with the circulating tumor DNA, like you're talking about, a lot of these companies talk about, very late stage patients and trying to do therapy selection, even though I will, caveat emptor that less than probably 5% of the time they're really actionable, with drugs that actually work. But, we offer those tests too. I think, the real challenge is, because on the disease side, the signal is high. So we announced the colorectal cancer, and in fact, we will be presenting at the ASCO, or at least our collaborator will be presenting an ASCO GU in February in San Francisco, where we will show results of the same platform for prostate cancer. So not to digress, but the problem, actually, the PSA has done a great job, even though it's much maligned, because it is a cheap, affordable test and the compliance is high. However, for men who have PSA levels between 4 to 10, maybe even 3 to 10, there's a lot of false biopsies. Like numbers could be 4 out of 5 or 1 out of 8. There's a lot of the PSA gives a false signal, so you end up getting a biopsy when you didn't need one. And the biopsies in the prostate, quite honestly, can sometimes be worse than the cancer itself, right? Because they come with a lot of complications. So there's a lot of motivation to find a test in that range that would be highly sensitive. And again, the PTC markers in our study have been highly sensitive. So one of the things I like to point out is that the compelling aspect of this platform is that once you can pick up the low number of CTCs, then it is applicable to all tumor cancers, and we've already started studies on breast cancer, etc., because it's the same mechanism. You're not looking at a fundamentally different technology to find these signals in various tumor cancers.
Tell me about the colorectal cancer study, though, and what it showed, and how accurate the test was.
So the overall accuracy of the test is in the high 80s. It's again a simple blood test, but the key finding was the ability to detect early stage cancer and pre-cancer lesions. And as far as we know, the fact that it's a blood test and doesn't look like there's anything else out there, and it's higher in accuracy than comparable stool tests, both for early stage as well as more importantly for pre-cancerous, right? So the results of that study were very exciting. We're continuing the studies, and we're going to do a study in the U.S. studies in the U.S. this year as well.
And why colorectal cancer? What makes that an attractive target for this technology?
Well, so like I said to you, that the effort started out originally a few years ago, in Taiwan, and so this is a very interesting statistic, and I think it's very informative that in Taiwan, colorectal cancer has been the number one cancer for the last 15, 20 years, as long as they've been sort of tracking it. And nobody, as with most things in cancer, nobody really knows why. And the reason, Danny, that this is important is because it's very instructive in general. Taiwan is a small country of 20 million people, so we get to work very closely, with their high-end hospitals. But they also have the best sort of socialized medicine in Southeast Asia, right? So literally the colonoscopies, the stool tests, for the most part, are free or highly, highly affordable and cheap. And even then, okay, the colorectal cancer goes up every year and has been the number one cancer. And more importantly, the compliance with stool test testing and colonoscopy is less than 33%. And the reason is, most people are reluctant to take a colonoscopy and, equally or more reluctant to do a stool test. So it goes back to the comment on compliance. So for as a starting point, it seemed to be, there was a lot of motivation in the hospitals there to look at colorectal cancer.
And if successful, how might this change the way that people are screened for colorectal cancer?
Well, as a innovative company, our answer is, that people keep doing what they're doing, but they also take, this blood test. Now, I can tell you again, maybe anecdotally, but of course the numbers are there too, that when you talk to people about taking colonoscopy, which is, the sort of screening test. And the reason, by the way, colonoscopy works as a screening test is because colorectal cancer is, relatively speaking, a slow-growing cancer. And therefore, the theory is that if you monitor it every, let's say 5 to 7 years, you're going to be okay. But the reality is that is not the case, right? So if we don't recommend people stop doing what they're doing, but they also start taking a simple blood test. And depending on their own assessment of the risk, an affordable blood test, they can take it every year, they can take it every 6 months, or they can take it every 18 months. So it makes it a very convenient additional, signal or indicator. And, if the test is positive, then they would be referred to take a diagnostic colonoscopy. And they would be, in our opinion, more motivated to take a colonoscopy and more likely to be found earlier.
And what's the regulatory path approval? Do you need to do additional validation? Do you need to get some kind of FDA sign-off?
Well, technically, the only to offer the test in the U.S., we really just need a CLIA, and then it becomes a question of affordability. But we are automating the instrument for CTC, and we do intend to decentralize it and have, partners, in the U.S. and all over the world. So we will be looking downstream at 510K for the instrument. But we, like I said before, we don't want all of those things to prevent the test from getting to the market. And the key issue, really, more than regulatory, which we can do with CLIA, is affordability, right? So the reason a lot of these tests get stuck is because, they have to wait for insurance reimbursement, which takes a long time. And the price points we want to come in at, we won't need that.
And what kind of price point are you at relative to other types of liquid biopsies?
Well, so again, I'll separate the liquid biopsies on the disease side, okay, where, like I pointed out, you have to do ultra deep sequencing because you're using ctDNA, and, it can be thousands and thousands of dollars, and it is. On the early detection side with our proprietary CTC platform, our goal is, by the end of the year to be at sub $150.
And looking ahead, what does the rest of your pipeline look like?
So, on the early detection side, clearly, we're excited with the capabilities of the platform, and we are extending it, and we want to accelerate that into other, tumor cancers like, prostate, which I discussed earlier, and breast cancer and lung cancer. I think there's also going to be interest for this platform to be used, by pharma companies. And as I pointed out, We already are selling our tests across the board in, about 20 countries. It's not, huge volumes, but so we want to be a global company. We think we understand, especially Asia, very well, where there's, 4.4 or 4.5 billion out of the 7.3 billion people in the world. And it's a different dynamic there and, on price points. So for completeness, we offer, for example, the hereditary cancer DNA risk test, which I will submit to you is the broadest sort of panel in the industry at the lowest cost. And on the disease side, we offer, like you were saying, the monitoring test. Now, on the monitoring, I will say one more thing, Danny, that, again, these liquid biopsies based on ctDNA that cost like $6,000, you cannot, the system, forget the patient, the system cannot afford those tests to be used for monitoring of recurrence, every 3 months or every 6 months. I mean, the whole thing will go bankrupt, right? So, but, with the CTC is using the same platform, essentially the same technology, and, we can do what's called a pan-cancer monitor test, again, for a few hundred, dollars that can be used as an additional signal for monitoring for recurrence. So, we also want to do the longitudinal data collection from people that come in, take the blood test to know if they are at a higher risk of cancer, and then, of course, the disease detection with the CTC test we're talking about, and, and then downstream, unfortunately, if they do get cancer.
Atul Sharan, co-founder and CEO of CellMax. Atul, thanks so much for your time today.
Thanks, Danny. Appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.